Hi, Graham.
Hi.
How are you doing?
Excellent.
It's great to have you. You are an algal biologist, currently associate professor at Colorado State University.
Yes.
And so you told me you're interested in understanding what proteins and processes are responsible for making microalgae successful in the world. So why did you decide to become an algal biologist, and why did you decide to study microalgae, basically?
So I thought my path was fairly random initially.
Mm-hmm.
So I started my career as an oceanographer, and I didn't really know what oceanography was. And my university had not a career day, but like a choose your major day. And there were all these people like crowded around like the marine biology station, right? And so there were marine biologists talking about, walruses or what have you. And I was like, oh, I'm not going to go. And sitting alone at a table was a very large man with a bow tie and thick glasses and an army haircut. And so I decided I'd go talk to him. And I don't know why. I was feeling friendly. And it turns out he was running the oceanography program at the university I was at. And he was very brusque. He said, don't take my word for this being the best program here. It's like, next week, the undergraduate students are presenting their research. There'll be pizza, there'll be beer. I'm from Canada, so drinking beer at undergraduate things is okay. Maybe not anymore, but at the time it was. And so I went, and I was just frankly blown away by the concept of oceanography, about thinking about biology not as an individual organism, but as a member of the physical environment, the chemical environment, and how all the three things actually all interact with one another to produce life that we see on the oceans. Mm-hmm. And that's what got me started. And I just happened to be more interested in the plant algae side of things than animals. Mm-hmm. I had a lot of friends who were further up in the education on animals side and doing not nice experiments with animals, and I never wanted to do that. Oh, so that's how I kind of got into that. So I thought it was this random thing, and then once I got my PhD, my mother reminded me that when I was very young, I used to like to go to ponds and pick up the algae. And
already?
Yeah. So she said, yeah, you actually showed a very early interest in the algae stuff. So I guess it was always there, but I don't know. Yeah.
Was that when you were young? Was that because they were microscopic green stuff, or did you know already that they were doing photosynthesis and fixing carbon?
No. I never thought about that aspect. Mm-hmm. When I was young, it was definitely through learning about things, maybe in my geology class and then other things, the importance that algae actually have in the environment, and always have had in the environment, building the atmosphere that we now breathe and things like that. It just became fascinating to me. And then I kind of like rooting for the underdog. I like working on algae because they're not plants. And people ignore them, but they're extremely important.
They are extremely important. So how did you get to focus on photosynthesis in algae at some point? Was it also random?
No, that one came about. I think that one came about in a low, slow thing. For my PhD, I was studying why the open ocean, why there's so few algae growing up there. So one of the pictures that someone sees of the ocean, open ocean, I mean, is like it's blue and you can see really deep, right? And the reason that happens is because there's no algae out there, right? There's just very little amounts compared to like when you go to the coastal regions, it's very dense with them. And we were trying to figure out why that was, and it turns out that there's not enough of one particular nutrient out there, and that's iron. So in iron, you and I use that for metabolism, right? Cofactor and enzymes. And then algae use that for photosynthesis, primarily. Most of the iron they use it for photosynthesis. So in order to learn more about the ecosystem, I also had to learn more about the biochemistry behind what the algae were using that iron for. And as soon as I started learning more, that's what I wanted to think about. I was no longer thinking about the cycle of carbon and things like that, but more about the nuts and bolts of how photosynthesis works.
Mm-hmm.
So
And
that's how I got to
so you started closer to the ocean, but then you transitioned to another microalgae system, which is a terrestrial system.
Yeah.
How did that happen? Why
So
you wanted to
I
enlarge your...
went from kind of like a bucket and boat scientist, right?
Mhm.
And I started reading more about how we could manipulate the DNA of organisms to really get at that mechanism, like this is how it works. And at that time, the only systems where that really worked well were this algae system called Chlamydomonas that you work on. And it was the only system where you could use the tools to get down to that molecular level to say, this is actually how it works, rather than just saying, we think maybe this is how it works.
Mhm.
And so I decided that I wanted to get experience doing that. And luckily my postdoctoral supervisor was willing to take a risk because I'd never done anything like that before.
Uh-huh.
Yeah.
It worked out
That's
pretty
so,
well.
so far so good.
Yeah, so far so good. So what are the I know you've switched from many different microalgae systems during your career. What are the systems you're studying right now? What are you most excited about? And what are the kind of tools that you're using to study their photosynthesis, basically?
Yeah. So my favorite organism to work on right now are diatoms.
Mm-hmm.
And
Is it because they're beautiful?
I do personally find them beautiful.
Mm-hmm.
Although to see their beauty, you need an electron microscope. So,
Oh, that's right.
well, I think just regarding them is beautiful.
Uh-huh.
But yeah, to see their true beauty, and one of the reasons, right, they're beautiful is because they make glass walls. So they make glass cell walls that are very intricate looking, but really most of the time you look under the microscope, they just look blurry, right? Because
Yeah.
most of them are very small. But despite the fact that they're very small and very blurry, they end up being probably one of the most important components of the food web in the ocean. So it's estimated that 1 in every 4 breaths that you take, whether you're sitting here with me or watching this online, is generated from a diatom. So they produce about 25% of the oxygen in the world. So, and then that also means that they fix about 25% of the CO2 out there. And they're very interesting. They're not plants, right? And I always want to stress that is like, they do photosynthesis, but they're as different in their evolutionary history from plants as plants are from animals. So there's this grouping of organisms, right? They're just so different in the biology. They make glass houses. They do photosynthesis in the water. Some of them can literally, they're unicellular, but they crawl. Okay, so you can actually watch them crawl along the microscope.
It is fascinating, yeah.
Yeah, and it's just it's almost wide open biology. So once again, think about that. A plant, an animal, what's the most different thing that we experience in our biological world? And there are things out there that are even weirder.
They're still doing similar, they still have similar photosynthetic reactions, right?
Yeah, their cores are the same. So they still evolve oxygen, they still use the same biochemistry to fix CO2, but there's some differences around them.
Mm-hmm.
And that's one of the things that we've been working on. So diatoms are brown in color, right? Just like seaweeds can be brown in color. Mm-hmm. And one of the things that we've been trying to figure out over the last almost decade is how they make that brown color. So the brown color allows them to absorb wavelengths of light that plants can't use, primarily green light. And we've known what that compound is for actually almost 100 years. So very early in biochemistry, people figured out what the color was, but we have no idea how it was made.
Yeah.
And so now we're going through those steps of figuring out how that compound.
So what are the tools you're using to interrogate that? Do you generate mutants?
Yeah, so we generate mutants using CRISPR-Cas9,
Mm-hmm.
which allows us to target very specific genes. So So we can say, we think that this particular gene is involved in the biosynthesis. And so we get rid of that gene. And the cool thing is, when it works, the diatom goes from brown to bright green.
Mm-hmm.
And it was shocking to us the first time. We thought we screwed up. We thought we had mixed two different things in the lab. Because we also grow green things.
Yeah.
Right? So like the very first thing I was like, oh no, we contaminated it. And so look under the microscope. No, it's real. Yeah, it was really cool.
And they're still doing photosynthesis.
They're still doing photosynthesis. They just don't do it as well.
Mm-hmm.
Yeah, they really do need that brown pigment to grow. They can live, but
Yeah.
they don't grow fast.
Does that mean that if we take these brown pigments, we put that in a green plant, would they grow better? Has that been tried?
Do you know, initially I would have told you no.
Mm-hmm.
Like, I don't think that's going to be possible because the pigments are actually need to be arranged very carefully to
Yeah.
work,
Mhm.
right?
Yeah.
So you need that happening. But what we've found is that these other pigments, the ones that kind of are in the pathway towards the brown, they work. So they actually are absorbing light and transferring that light energy to chlorophylls that ultimately lead to photosynthesis. So they work, they just don't work as well. So this leads me to think that it might actually not be hard to transfer that brown pigment over into an organism that's green.
That would be interesting.
It would be. And so, like I said, initially I was like, this would never work. But now,
Yeah.
now that we're looking at it,
And with all the developments in parallel with the introducing chlorophyll F or other types of chlorophyll in sand bacteria or in green algae,
yeah,
probably will become a reality soon.
yeah.
Maybe.
I think we'll be, I think, you know, maybe not reality, but we will be able to try.
Yeah, exactly.
Yeah. Yeah.
And to see whether it's able to generate photochemistry
Yeah.
in other organisms. That's pretty cool.
Yeah.
What are the tools you're using to, once you've done your genetic modification, what are the tools you're using to probe photosynthesis or the fitness of algae? Are you growing them in photobioreactors,
Yeah,
plate?
yeah, we do a little bit of everything. I think one important thing that we focus on in our lab is ecophysiology, of really thinking about, like, we don't want to uncover something that's just important in the lab, right? We want to know whether this is important for the organism in its natural environment, right? And so we try our best to like do several steps, controlled steps, to say, okay, you know, are they better able to absorb low amounts of light? Do they saturate faster in high light? But then we want to take, so once we come up with that yes or no, then we want to transfer them into something more realistic. Mm-hmm. So we simulate light environments that you'd find naturally in the ocean, in the lab, and then we'll grow them in the lab and see, you know, how they respond. Do they grow slower, faster? And the answer is, yeah, they grow slower. Yeah. Not surprising. But,
Makes sense.
But still, you'd be a little shocked if they did grow faster. You'd wonder why.
Yeah.
Yeah.
Is there a recent discovery in your field that you found really exciting and that you feel is driving the understanding of this algae and photosynthesis? Maybe in diatoms, maybe in other systems?
I think it's not necessarily one step, but I think what we're becoming more and more aware of is of the diversity of ways that plants and algae solve the same problem.
Yeah.
Right? That now that we're being a little more aware of the diversity of organisms that are out there, the different environmental stresses or evolutionary paths they've taken, I think the field in general is saying, well, it doesn't have to be like a plant. It doesn't have to be like corn or something like that, which grows and does photosynthesis very well. Right? There are many other ways that organisms have figured out to do photosynthesis well. And I think that's what's really exciting and driving our field right now. Yeah, is that open-mindedness.
Yes. So where do you see that open-mindedness taking the field in the next 10 years? Will we just discover, like, be stamp collectors with just, okay, these are the very different ways, or will that come up into like a more broad general understanding of what are the strategies that make photosynthesis efficient? Or
so to use your analogy of stamp collecting, I just made this up by the way, if you don't have the stamp, other people aren't going to get the message, right? So you need that stamp initially in order to send out the information, right? And I think this is what's going to happen, is we're going to find maybe simpler solutions to complex problems that allow us to then take that information and apply it to things that are important for us in society, so crops. Right? It could be crops, it could be growing algae for high protein feed, all these sorts of things. I think, we got stuck in the area of plant biology of having to solve plant photosynthesis using a plant model. So the evidence is trying to make C4 photosynthesis in a C3 plant. It's carbon concentration. But it turns out there are all these other ways of doing it without invoking very complex changes in structures within the leaf and things. So I think that's going to be the really exciting aspect.
Being able to transfer from different organisms, different processes that could be easier or possibly, more efficient, maybe.
Or possibly just seeing a pattern. Right? Like this, when we see this happen, and you don't even need a mechanism necessarily. Oh, look at that, that's really cool, that's different. Maybe if we can find a plant that does something the same way, we can figure out how it does it and then apply it to crops. Like I said, it's the open-mindedness now that's really exciting about it.
So what do you feel the impact will be for society? Will it be only for crop plants and maybe for algae for some protein production or pigment generations?
Well, I think when we think about crops, we have to think it's not just about food, right? So photosynthesis, right, at best in a crop plant, 0.5, 1% efficient, right? But imagine that you improve that, you double that, right? So from 0.5 to 1. Well, if you can do that, that might mean that you need half the amount of fertilizer, that you need half the amount of water.
Half the amount of land?
Half the amount of land, right? So what seems like a very small number, right, and insignificant, actually translates to major things in the end. Like, if we could use half the amount of water to irrigate our crops, that's going to have a major effect in California, Nevada, everywhere else, places that are approaching big trouble. So I think that's one of the exciting things about photosynthesis, is it's not just photosynthesis, it actually will impact many, many other things.
Anything that's using biology eventually. Cool. Thank you very much, Graham, for talking to us.
All right. Stay tuned next time for two bald guys with glasses talking science.